Welcome, everybody. Thank you for joining us for the live and in-person recording of Editors in Conversation. We're happy to see all of you. As introductions, I'm Ellie Thiel. I'm one of the editors for the Journal of Clinical Microbiology, and I co-host with my co-host, Dr. Alex McAdam, the JCM Editors in Conversation. Dr. McAdam is editor-in-chief for JCM, and he works at Boston Children's Hospital. Alex, I'm told that you have a question for the audience.
I do have a question for the audience. Can anyone in the audience, there's a prize, can anyone in the audience tell us the next new journal that ASM will be publishing? Back there, nice and loud. Yes. ASM case reports.
I will throw you the shirt over here so I don't mess up the lighting.
Can you hold it up?
Oh, yes.
Yeah. ASM is launching a new journal, ASM Case Reports, and just this week, the editor-in-chief for the journal was approved. It is our good friend, Dr. Carrie Ann Burnham. Carrie Ann has been an editor of JCM for 10 years. She was coming to the end of her time at JCM, and I could not be happier that she is taking the helm at ASM Case Reports.
I agree. I think it's going to be a great start to a brand new journal. Okay, so getting into our podcast for today, if you listen to the podcast, we usually like to have some fun. When we do the live recording, we like to have more fun. And so this year, we decided that we're going to talk about headlines in the news that had to do with infectious diseases or microbiology. So these are all headlines pulled from the news. And as part of the fun, we have audience participation. So you get to answer the question we have. But then we also have two experts, clinical microbiology experts. They're going to talk to us about the accuracy of the headlines and how concerned we need to be about some of these headlines, and also whether we should be believing the headlines. So without further ado, we have a repeat guest, Dr. Ben Pinsky. He is the director of the Clinical Virology Lab and a professor of pathology and medicine at Stanford Healthcare and Stanford University School of Medicine. Thank you, Ben, for coming. And then we have new to the podcast guest, Dr. Greg Berry. He's an associate professor of pathology and cell biology and co-directs both the Clinical Microbiology Service and the Center for Advanced Laboratory Medicine at Columbia University. Thanks for joining us, Greg. Okay, so we are going to get to the first headline. So here's your first question. The USDA recently started testing ground beef for what virus? Thoughts? Well, H5N1 is the answer. Nod of heads. Who agrees? Most everyone agrees. Very good. That is correct. So we're going to turn to our experts. Ben, can you bring us up to speed on where we're at with H5N1 cases? Where are these cases occurring? Tell us what you know.
All right. Yeah, absolutely. Thank you. Thank you both for inviting me back. Apparently, I wasn't so bad last year.
You're all right.
So H5 avian influenza virus is of concern. Over the last several years, it's come to the United States, a particular clade 2344B. You don't have to memorize that. And primarily in birds and farm poultry, wild birds and farm poultry. Over the last several months, this virus has infected dairy cattle or dairy cows, and that's been of significant concern. It's the first time the virus has been identified in that species. And this virus is supposed to stay in birds, but when it goes into mammalian hosts, can then adapt. And we get worried that it may cross over into us, into humans. And there have been a number of cases of human influenza A H5 over the last couple of years. The first was early on in this outbreak where an individual who handled ill birds got sick in Colorado. But subsequently, over the last few months, there's been three human cases in dairy farm workers, and these folks were exposed to cows that were sick. The first two cases were quite interesting in that they primarily presented with conjunctivitis and did not have significant respiratory symptoms. However, and those two cases, I should say, were in Texas. One in Texas, one in Michigan. The third case, most recent case, that individual, also a dairy worker, did have respiratory symptoms. So this is becoming a little bit more concerning for human spillover events. There's a lot of monitoring at the moment, as mentioned in the question of beef, where viral RNA at least has been found by report. In addition, the virus has been found in pasteurized milk products, the viral RNA. However, the virus has not been cultivatable from those samples, so does not appear that pasteurized milk is infectious.
So you mentioned pasteurized milk and beef. What about raw milk? Is that a potential for spreading the
Yeah,
virus?
so that's an important point. Raw milk is a potential source of human infection. There's a very nice article in Emerging Infectious Diseases describing the outbreak in dairy cows and also an outbreak in cats, barn cats, that were fed unpasteurized milk from these sick cows. And cats are apparently very susceptible to severe avian influenza infection. And I think about half of these cats that were fed this milk died with disseminated H5 influenza A infection, including viral inclusions basically in every tissue, but they had severe neurological disease with virus detectable in the brain.
Another reason to drink pasteurized milk.
Right, so don't drink pasteurized milk. Fortunately, our influenza A assays should detect influenza A H5. We just won't know the subtype. Right now, where there's low influenza activity, we should be able to identify if there is an outbreak in a spillover event. And of course, there is enhanced surveillance of dairy farm workers, of wastewater. So hopefully, if this does spill over, we'll be able to identify it rapidly. If unfortunately this were to happen during respiratory season, things would get a little bit more complicated and we'd need better H5 subtyping tests if that were to be the case.
Thank you, Ben. We'll move on to our next question for the audience. What state recently had a case of locally acquired bubonic plague? Any takers? Yes. Very good. The answer was Oregon. That is the correct answer. And Greg, we're going to bring you into this.
Sounds good.
So talk to us about this Oregon case. How is it transmitted in this particular case? And how many cases of Yersinia pestis do we usually have in the U.S.?
Yeah, so this Oregon case was an interesting one because it appears that the person who contracted the disease actually got it from their cat. So their house cat somehow had been exposed, and I could go over how we think the cat was exposed, but yeah, they ended up confirming that the cat actually was carrying Yersinia pestis. And so while you think of Yersinia pestis as a disease of antiquity, it's still around, it's still endemic in the population of rodents out west. You still see it in chipmunks, you see it in squirrels, and you could see it in mice. And so the thought is, well, really how bubonic plague gets spread is through flea bites. So the thought is that perhaps this cat picked up a flea that was on a chipmunk or a squirrel or some other rodent that was actually carrying the disease, and then subsequently brought it into the house, and then the owner ended up getting bit with a flea and contracted the disease. So, and the prevalence is like about 5 to 10 cases a year, so it's not that prevalent. But actually in Oregon, this is the first case since 2015.
Yes, and this is another reason I don't like cats.
I was going to say that, but I wasn't sure if I was allowed to, so thank you.
You're allowed. So, we all know about the bubonic plague and how it killed over 50% of the population in some regions of the world. And that ended up leading to some genetic evolutionary pressures. Some of those were published in Nature in 2022. And can you tell us about that paper and what those genetic pressures were and how kind of humanity has evolved as a result of the bubonic plague?
Yeah, in fact, if you think about a disease that can kill 50% of the population, it literally is a genetic bottleneck, right? It had the ability to actually influence changes in the human genome moving forward because survivors presumably had some genetic protection against the disease. And in fact, the study that was in Nature did show that there was two different genes. One in particular actually showed 40% protection from Yersinia pestis disease. And so while that protective mechanism was in place and subsequent generations, it could be found in subsequent generations currently present day in London and in Denmark, there are also unintended consequences of that particular mutation. It's not such a surprise that a mutation like that would also carry other effects that do not necessarily influence Yersinia pestis, but have other off-label effects. And in this case, what it is actually a predisposition to autoimmune diseases, and specifically, I forget if it was ulcerative colitis. I believe was the actual. It was ulcerative colitis. So,
Thank you, Greg. And lastly, there is a word that we use a lot during the pandemic, and it actually originated during the days of the Black Death. That word is quarantine. Can you tell us how that word came about and how it was associated with Black Death?
yes, absolutely. So again, back to a really old disease. There's a lot of history here, and when you think about isolation from diseases in general, this is millennia back that people would, if they were potentially sick, there were isolation measures in place. This is back to biblical times when you think about leprosy. But the first use of the word quarantine apparently comes from Dubrovnik, Croatia. So Dubrovnik was a wealthy trading city. And at the time, during the Black Plague, they were trying to come up with ideas to keep business running, but also not infect the whole city. It's quite comparable to what we just went through with COVID, right? Trying to figure out measures to stop spread of infection without killing the economy, right? So they came up with the idea that if people were going to come to the city and they were going to trade, they had to spend 40 days outside of the city before they were able to come into the city. And there was some really draconian measures in place to make sure that they followed that particular mandate. So the word quarantine actually comes originally from Italian. So the words for 40 days in Italian is quaranta journey. Quaranta journey is 40 days, which turned into quarantino, and quarantino is present day in English quarantine. So
I love that you brought in the accent. That was really good.
I am so happy right now, Ellie. Thank you for
Grazie.
including that question. Awesome.
That was very, very good. Yes. All right. Next topic. Question for the audience again. Probably won't be too difficult, but which vaccine preventable disease is having a comeback, maybe again, in the United States? Any takers? You're going to have to yell a little louder. Measles. Do we agree with measles? Yes, we all agree with measles. That is the correct answer. All right.
So, Ben,
I'm sorry.
Did you have
Go
a
ahead,
question,
Alex.
Ellie?
I do not have a question.
do you want to borrow my script?
No, I don't. Even though I wrote it.
Sorry, I stepped on your line. I'm sorry. Ben, why and where are we seeing measles cases spiking? And how does it compare this year to last year for the total number of cases?
Yes, so measles has been resurging over the last several years. In the United States, already this year in 2024, there's been over 150 cases compared to the entirety of last year where there were 58. So it's about 2.5 times, I think. I do the math right.
Very good job.
Okay. And that actually, that's a lot for the United States, but in other parts of the world, we're seeing enormous numbers of measles cases. Last year, there were over 300,000 cases worldwide. And so far through through this year, there's been over 90,000 cases. Many of those have been in Central Asia, where there's been huge outbreaks. And a lot of this has to do with under vaccination with MMR, measles, mumps, rubella, live vaccine. The numbers of vaccinated individuals or percentage of vaccinated individuals, actually children at 1 year of age, pre-pandemic was about 86% globally, and that's gone down, I think, to 81%, might be bumping back up. Ellie made me look up all these numbers, so hopefully those are somewhat right. The bottom line is that it's gone down globally, and there's under-vaccinated populations. In the United States, our vaccine rate has gone down. This is at a different time frame, kindergarten-age children, to 93%, and our goal is 95% to have effective herd immunity to protect the population. Sorry, you want to ask a question, or can I go on?
No, please go on.
Most of the cases in the U.S. have been travel associated, individuals traveling to places where there's large outbreaks, and coming back and exposing a susceptible population. Some of the largest outbreaks in the United States have been in the Chicago area, in migrant shelters, mostly individuals seeking asylum from Venezuela. So there's been large outbreaks there. And as you all know here, measles is amongst the most transmissible of infectious diseases, with a secondary attack rate of over 90%.
Thanks, Ben. We did give you all the hard questions with all the numbers you had to memorize. And I have one more question with a number. Can you tell us how many cases constitute an outbreak?
Yeah, I did have to look that one up too. So an outbreak is defined as 3 or more linked cases. So it's a relatively small number of individuals.
Awesome. Thank you, Ben. Oh, did you have a question?
I did not.
What? Yes. Oh, I didn't know we could.
We have measles on the table.
Give
somebody measles.
Give the crowd measles.
I mean, hopefully we're all
Very
vaccinated. All right.
Who answered our measles question?
A deal.
Pass it back. In the front.
Okay.
Thank you.
So last question, Ben, on measles is, in the U.S., measles was eliminated in the year 2000. Can you remind us, what are the criteria that a country has to meet in order for us to say that measles has been eliminated in that country?
Right. So it's there cannot be endemic spread over a time frame of 12 months. So that's the definition. We're having an increase in cases. It doesn't seem like we're at a risk of losing our status. In the years before the pandemic, there was a year where there were over 1,200 cases in the United States, primarily, again, in under-vaccinated communities in New York State, due to, again, import from other countries, but then exposure to a large number of individuals that had not been vaccinated. So it's another advertisement for vaccination. It's very effective.
Great. Thank you. So next topic. Which eradicated mosquito-borne pathogen was recently locally acquired in Florida and Texas and Maryland? And if you were at the literature review, you should know the answer to
There's
this.
a hand up in the back.
Oh, yes. Malaria. Very good. That is correct.
Greg, let's turn back to you. How many cases of locally acquired malaria have we had this year, and what species are we seeing?
So yeah, currently there's been 9 cases. There were 7 in Florida, 1 in Texas, and 1 in Maryland. The lion's share of the cases were Plasmodium vivax, but there was actually 1 case of Plasmodium falciparum that was in Maryland.
Thank you. And I think Ellie wrote these questions just for me, because the next one is about history, which I love. Can you remind us, Greg, when malaria was eliminated in the U.S., and how long it has been since the last time we had locally acquired cases?
Yeah, so It's actually not that long ago. I think people think that malaria has been eradicated in the United States like over 100 years ago. Actually, you come right up to the 50s, the 1950s in the United States, and malaria was still considered endemic. In fact, endemic until the year 1951. And then we saw, it's been now about 2 decades since we saw local transmission. So we had a big gap of 2 decades, but now with local transmission with 9 cases, it's concerning.
So, since we don't have malaria that's endemic here, can you tell us how these people got infected and how worried we should be about malaria outbreaks?
Yeah, so while I would say that seeing a trend of 9 cases is concerning in the sense that we don't want to see malaria in the United States, it's not really a cause for alarm at all, because what most likely happens. So to give it context, we see about 2,000 to 2,500 cases of malaria diagnosed in the United States every year. But those are diagnosed in travelers who come back to the United States infected. Well, in these cases, what happened is somebody came back after traveling infected, got bit by an Anopheles mosquito, and then basically that mosquito got infected and was able to bite somebody else and start the chain of infection. But for that to occur, it's pretty rare. It's pretty rare. And then on top of it, I mean, if you think about that, it has to be a traveler who gets bit by a mosquito. For local transmission to actually start and initiate, you would have to have more widespread infection and start that cycle. So it would be quite difficult for that cycle to start.
That's very comforting. Thank you. All right, last topic. This is my favorite. This is my favorite one. Okay, so I need the audience to complete this sentence. A study showed that rubbing Neosporin here, so which body part, can decrease your risk of getting these sorts of infections, meaning bacterial, viral, parasitic. So, anybody want to fill in the 2 blanks? Have fun with it. Nobody? Yes. 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 So the body part and what type of... Oh, okay, good.
Very
That's
good.
half correct. Very good.
Which part was correct?
The nose.
Nose.
The nose was very good. And then...
It should be
anybody
staph.
else? It should be.
You said staph, and that
Yeah.
should be the right answer.
But that is
But
not
it's not.
the right answer, which makes it even more fun. Should we tell him?
Yeah.
You know the answer, Greg. All right. So this is the headline that caught my eye. Can Neosporin in the nose help prevent viral infections? So Ben, you're a virologist.
They say.
So they say. What was your initial reaction when I sent you this headline?
I was a little bit confused, but then you sent me the article,
Yes,
which is quite interesting.
Tell us about the article. Walk us through it.
Yeah. So in this study, I believe out of Yale, and I'm blanking out on which journal it was in.
Not yours.
Not mine. For sure, much a higher profile. They showed that Neosporin caused the upregulation of genes involved in the interferon response. And so in sort of a semi-indirect mechanism that these increased interferon response was protecting in cell lines and in mouse models, and in a actually, I don't think they did a human protection trial. They just showed that the antibiotic could increase these transcripts in humans. So it seems like there's a plausible mechanism that this antibiotic could potentially protect us from respiratory viral infections, including SARS-CoV-2.
Thanks, Ben. So I have to ask, Greg, will you be dabbing Neosporin in your nasal cavities this respiratory season?
I mean, this is just my opinion and not the opinion of JCM, ASM, or any other organization. But no, I will not be, because I'm worried about antibiotic resistance. So I'm going to opt out of that option.
Alex?
I will also pass. I think there are many other things we can do to prevent respiratory viral infections, like get vaccines, practice hand hygiene, stay home or mask if you're ill, and that would put Neosporin low on my list.
Excellent. I think if I went up to my child and said, "Let me put something in your nose," after their COVID experience, they would be running. Awesome. Well, those were all of our headlines for today, but
There's more.
there's more. Oh,
We have a few limericks. Now, you have to let me read through the limerick, and then you guys are going to work together to provide the last line or last word of the limerick. And this is shamelessly stolen from a popular NPR quiz show. So let's have our first limerick, please.
And they have not seen these,
They
so
have
they
not
don't
seen
know the
these.
answers. Let's see.
Our first one is a limerick. Nothing? Okay. Canines get shots of OSP-A, but people don't. Is that okay? A protein vaccine disappeared from the scene. The new shot has...
The barn. Ah.
What do you think?
It's mRNA.
mRNA.
very
That's exactly
good.
right.
Ding, 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 ding.
Ding, ding.
Very good. So this was published by Dr. Norbert Party and colleagues out of UPenn in molecular therapy. And just as you suggested, Greg, they designed and tested an mRNA vaccine with OSP-A, and it stimulated both humoral and cellular immunity in mice, and it protected the mice from infection with Borrelia burgdorferi. Well done. You're 1 for 1. Let's have our next, please.
Good work.
More about the plague. A bubonic plague isn't nice. Avoid it. That's my strong advice. This awful disease is spread mainly by fleas, but a second vector is...
Mice?
No.
No. Lice.
Lice. Yes, that's correct. That is correct. So this was published in PLOS Biology by Dr. Joe Hinnebush, who is at the NIH-NIAID. And his group demonstrated that the body louse, Pediculus humanus, can become chronically infected with Yersinia pestis, both in the GI tract and in accessory salivary glands, which are shown there in red, suggesting potential routes of transmission via defecation or feeding by the louse.
I like
All
that
right.
you added maybe there, just so you're not held to it.
It's maybe. They didn't actually demonstrate transmission.
Perfect.
All right, our last one, 2 for 2. Let's go for 3. And we have another plague. Plagues come in many versions. Not all of them occur in persons. This one's in the sea. The victims are spiky. This plague is killing off.
Urchins.
Urchins is right.
Wow. Very good.
So this is a paper, well done. This is a paper by Dr. Bronstein, which was published in Current Biology, and it describes the die-off of sea urchins in the Gulf of Aqaba in Jordan. This appears to be caused by a ciliated protozoan species that has been previously described to occur in the Caribbean and off the Florida coast in 2022. The earlier work was done by Dr. Henson and published in Scientific Advances. And in that paper, they fulfilled Koch's postulates for this pathogen. Well done, 3 for 3. Congratulations.
Well
Let's give them a hand.
Yes. Good job.
Next year, maybe some viral limericks. If I'm invited back.
I don't see it
Absolutely
coming.
not. They're all going to be
We will
serology.
definitely invite you back. I don't know about the limericks.
Well, thank you, everyone, for joining us. Hopefully you learned something, some history, some, depth to
Lousology?
urchins.
Urchinology.
Urchinology. And we hope you enjoy the rest of the meeting. Thank you.